Hi guys, welcome back to my YouTube channel. If you are new here, subscribe to be notified as soon as I upload a new solved past paper video with simplified explanations. Don't forget to like and comment. I'll be glad to answer any doubts. Alright, so let's get started. A student investigated the digestion of starch. Figure 2.1 shows the apparatus she used. Dialysis tubing is used to represent a cell membrane. The dialysis tubing material allows small molecules to move across it, but not larger molecules. Test tubes A and B were set up as shown in Figure 2.1 and placed in a water bath at 37 degrees Celsius for 30 minutes. The liquid outside the dialysis tubing in test tubes A and B was tested with Benedict solution at 0 minutes and after 30 minutes. Table 2.1 shows the results. Test tube A, the color with Benedict solution at 0 minutes is blue, and color with Benedict solution at 30 minutes is also blue. Test tube B, the color with Benedict solution at 0 minutes is blue, whereas the color with Benedict solution at 30 minutes is red. Using the information in Figure 2.1 and Table 2.1, explain the reasons for the difference in the results for test tubes A and B in Table 2.1. Benedict solution is a reagent used to test for the presence of reducing sugars. The solution changes from blue, indicating the absence of reducing sugars, to a red or orange color, indicating a positive result for the presence of reducing sugars like maltose. Benedict solution in A remains blue as there is no maltose. Benedict solution in B turns red due to the presence of maltose, indicating a positive result. Amylase is an enzyme that breaks down starch into smaller, simpler sugars like maltose. Maltose, being a smaller and soluble molecule compared to starch, can pass through a selectively permeable membrane like dialysis tubing through the process of diffusion. Diffusion allows molecules to move from an area of higher concentration to an area of lower concentration until equilibrium is reached. Therefore, in summary, Benedict solution will remain blue, no color change, in solution A where there is no breakdown of starch into maltose. In solution B, where amylase breaks down starch into maltose, Benedict solution will turn red, positive result, due to the presence of maltose, which is a reducing sugar that reacts with Benedict solution. Additionally, maltose being a small and soluble molecule can pass through the dialysis tubing by diffusion from an area of high concentration, inside the tubing, to an area of low concentration, outside the tubing. Complete Table 2.2 by writing in the names of the missing enzymes, substrates, and products. Pepsin. Right, so pepsin is an enzyme produced in the stomach. The primary substrate of pepsin is protein, and the products of its action are shorter peptide chains and individual amino acids. These smaller peptides and amino acids are then further broken down and absorbed in the small intestine during the digestive process. Therefore, we can fill it as protein for the substrate and the products are either amino acids or peptides. Secondly, right, this time the products are given to us, fatty acids and glycerol. Okay, so fatty acids and glycerol are the components of fats and triglycerides. Enzyme responsible for breaking down these molecules into smaller components is lipase. Right, so the substrate can be either fats, oils, or lipids, and the enzyme is lipase. Next up, the enzyme trypsin. Well, trypsin is a digestive enzyme primarily produced in the pancreas and involved in the breakdown of proteins into smaller peptide fragments. Its main substrate is proteins and the products, therefore are amino acids or peptides. Okay, so the last one, now again, we're given the products and we need to fill the enzyme and substrate. So the product is glucose. Maltose is a disaccharide composed of two glucose molecules linked together. When maltose needs to be broken down into its constituent glucose units for absorption into the bloodstream, the enzyme maltase comes into play. Maltase specifically catalyzes the hydrolysis of the maltose molecule, breaking the bond between the two glucose molecules, thereby producing two individual glucose molecules. So we can fill it out: enzyme, maltase, and substrate, maltose. Final part of question 2, state the name of the structures that increase the surface area for absorption in the small intestine. The small intestine is adapted for efficient absorption of nutrients, and its surface area is significantly increased by intestinal villi or microvilli. The villi greatly increase the surface area available for absorption. They contain blood vessels and a lymphatic vessel called a lacteal, which help in the absorption of nutrients into the bloodstream and lymphatic system, respectively. Alright, so here we reach the conclusion of this video. I will be posting the next part shortly. You can visit the entire playlist, and if you have trouble with selective questions, you can watch accordingly. I try to provide detailed but easy to understand explanations so that everyone can follow along and understand the concepts. Thank you for watching. Please like if you found the video helpful and subscribe for more.